Okay, so today we're going to do some genetics, and I'm going to show you the difference between a complete dominance Punnett square, a codominance Punnett square, and an incomplete dominance Punnett square. So you want to do, you want to keep good notes with this so that you can do your practice problems. All right, so complete dominance is your typical Mendelian dominance, and it's where the capital A might stand for, let's say, black hair. And the lowercase a stands for red hair. And in this situation where it is complete dominance, we can be assured that this allele always beats this allele. So when you're crossing two parents and you're trying to determine their possible outcome of offspring, black hair, let's say that one parent is heterozygous for black hair. That means they have one dominant allele and one recessive allele. And the other parent has red hair. So I know for sure that their alleles are two little a's because red hair is recessive. And if they had a capital A allele, a dominant allele, they would not have red hair, they would have black hair. Then when you do the Punnett square, you take the options for parent number one. So parent number one could give the dominant black allele, or it could give the recessive red hair allele. This parent, really and truly, only has the red alleles to give, the recessive alleles. So then when you fill in your Punnett square, you're filling in the possible combinations of parent one with parent two. So parent one could give the big A, and parent two the little a. Parent one could give the little a, and also parent two. Parent one could give the big A, parent two give the little a, and then parent one give the little a, parent two give the little a as well. So when these two parents with the genotype a heterozygous for black, homozygous for red, have babies, you could tell them, okay, so black hair is a 2 out of 4 chance because it's that one, and red hair is a 2 out of 4 chance. Okay, so when these two people have an offspring, there's a 50-50 chance that it has black hair or red hair. Complete dominance works that way. You can always be assured that the capital letter will always beat the recessive lowercase letter. But when we do codominance and incomplete dominance, we have a different scenario. So I'm going to draw a line down the middle, and I'm going to do codominance on the left, and I'm going to do incomplete dominance on the right. Okay, so co means both. So one is not dominant to the other. They're going to both show up. And in science, we call this expressed. Okay, so when a trait is expressed, it's actually what you see. So physical attributes are called your phenotype. All right, and your letters that you get are called your genotype. And those are composed of alleles, right? And everybody has 2 alleles for every trait because you get 1 allele from each parent. Incomplete dominance is a situation where one doesn't win and dominate the other, and both of them don't show up. Incomplete dominance is when they blend. So it's a blend of the 2 phenotypes. All right, so I'm going to give you an example using similar characteristics and show you what it would look like if the gene was codominant and then if the gene was incomplete dominance. So every animal has different ways that these things work. But in codominance, I'll do big B is the gene for black fur. And you're going to notice that I'm not going to use any lowercase letters because there are no recessive traits. So everything I use is going to be capital. And big W is going to stand for white fur. Okay, and I'll let this be some sort of mammal. And I'm going to cross a black furred animal. So both of its alleles are going to be black with a white furred animal. Okay. And in a codominant situation, you would do the Punnett square. should be pretty obvious to you that this animal will get a black gene and a white, a black allele and a white allele from each parent. So in this situation, if black was dominant to white, all the offspring would be black. Or if white was dominant to black, all the offspring would be white. But in this specific scenario, we are getting 100% spotted. Okay, so we could say white with black spots or black with white spots. Doesn't really matter. It's just that they both show up. So both alleles are expressed. If this same trait had an incomplete dominance pattern of inheritance, let me write that at the top. These are called pattern of inheritance. Okay, and these are your two examples. So if it happened to be an incomplete dominance pattern, I'll use the same alleles. I'll use black fur and I'll use white fur. And if in this situation, I cross a black furred animal with the white furred animal, same scenario. The parent with the black alleles could only give black alleles. The parent with the white alleles could only give white alleles. So then the offspring, every single one of them, are going to be 100% this. That's going to be their genotype. Their phenotype, in an incomplete blend type and pattern of inheritance, they're going to be 100% gray. Okay? Difference between the two. If the genes have a pattern of inheritance of codominance, you're actually going to see both show up. If the pattern of inheritance is incomplete, you're going to see a blend. Okay? So, one more little example here. Codominance, incomplete dominance. All right, I'll give you two different examples. So I'll start with incomplete dominance. This happens a lot in flowers. So you'll have a red flower mixed with a yellow flower, right? And if all of their offspring are red, then it would be complete dominance. If all of their offspring are white with red spots or stripes or something, we would call it codominant. But if all of the offspring are orange, and if you didn't know, red and yellow mixed together make orange, this is going to be incomplete dominance. All right, so if you see these patterns happening, this is how you can determine what type of inheritance we're looking at. So in incomplete dominance, red flowers would have two red genes. Yellow flowers would have two yellow genes. There is no, alleles. There is no allele for orange. Orange is accomplished by giving an organism a red gene and a yellow gene. So if I asked you to cross an orange times an orange colored flower, you would have to start with the letters RY. Now, it doesn't matter if you put Y first or R first. This is your codominant situation. So an RY means a red with a yellow, so it makes it orange. And then you cross it with another RY, red and yellow make orange. Then when you do the Punnett square, parent number one could give the red gene to the offspring or the yellow allele. I got to stop saying that. It's allele. Okay. Parent number two, red allele, yellow allele. Then when you fill in the box, that offspring is going to actually be red. This offspring is going to get a red and a yellow gene, so that one, or allele, that one will be orange. This one's going to get a red and a yellow allele, so it will be orange. And then this one's going to get two yellow alleles, so it will be yellow. So in a cross in an incomplete dominant scenario, orange times an orange, you get 1 out of 4 chance that it's red. You get 2 out of 4 chance that they are orange. And you get 1 out of 4 chance that they are actually yellow. So these flowers will cross and reproduce. And if they had 100 seeds, you would expect 25 of them to be red, 50 of them to be orange flowers when they grow, and then 25 of them to be yellow, because that would be a fourth, and a half. All right, let's go over to codominance. Codominance, we're both showing up, okay? So I'll give you a different scenario, and we'll do yellow leaves, and then we'll do a plant that has blue leaves. Okay, so if we cross these two and we got 100% like blue, then we would know blue is dominant to yellow. If we cross these two and we got green, we would know that it's incomplete dominance, because there is no allele for green. It's a blend between yellow and blue. But if we mixed them and we got blue with yellow stripes or yellow spots, how about that? Then we'll know it's codominance, because yellow didn't beat blue and blue didn't beat yellow. They both showed up. So if you saw this and I told you that a yellow leaf plant mixed with a blue leaf plant makes a blue leaf with yellow spots, you have to know that's codominance. And you also have to know that your letters will not, there's not an allele for spotted. So you would have to do yellow plants have 2 Y genes and blue plants have 2 blue genes. So a spotted plant would be YB. That would be its genotype. So let's say I ask you to cross a spotted with a yellow. Then you have to know how to write its genotype. Then you have to know Punnett square. Take the first parent can give a yellow allele or a blue allele. Second parent can give nothing but yellow. All right. So when you cross these two, filling in your Punnett square, and you can put YB first or BY, it doesn't matter. You can say that if these two plants were crossed and pollinated and bred, then they would have 2 out of 4 would be spotted, which would be that one, and then 2 out of 4 chance would be yellow. Okay, and then it would be 0 out of 4 chance for blue. So if you cross these two plants and you grew 1,000 plants, 500, around 500 should be spotted, around 500 should be yellow, and about 0 should be blue. So these are the probabilities of the offspring of mixing certain alleles. So I'm going to give you a worksheet, and you're going to use these codominance and incomplete dominance to fill in the worksheet, and we will go over the answers on the next video.